The nucleus is usually the most prominent feature of a cell. Most body cells have a single nucleus. However, some cells, such as red blood cells, when they're mature and functioning, don't have a nucleus at all. Other cells, such as skeletal muscle cells, have multiple nuclei. The parts of the cell include the nuclear envelope, which is perforated or has holes. So these perforations that are called nuclear pores serve as an area for things to move out of the nucleus and into the cytoplasm. Then in the center area of the nucleus is a structure known as the nucleolus, and the genetic material, the DNA, is located within the nucleus as well. The nucleolus functions to help produce ribosomes. Each nucleolus is simply a cluster of protein, DNA, and RNA, and it is not bound by a nucleus. Within the nucleus are all the cell's hereditary units called genes, which are arranged on the chromosomes. Each chromosome is a long molecule of DNA that's coiled together with several proteins to give it structure. Human somatic or normal body cells have 46 chromosomes arranged in 23 pair. So you get one set of chromosomes from mom, one set of chromosomes from dad. So you get 23 in the sperm cell, 23 in the egg cell, and that gives you the total of 46 chromosomes. So here you can see the double-stranded DNA helix wrapped around these little proteins called histones, and then they compact or condense down into structures known as chromosomes. All of the genetic material collectively found in the human body is called the genome. So genomics, or the study of the genome and its relationship to body function, has the potential for increasing our understanding of normal and abnormal conditions related to those chromosomes. Because remember, when we first mentioned chromosomes and genes back in a previous video, we talked about those genes hold the recipe for making all of our proteins. So if a gene is mutated or it no longer functions as it should, then we may end up making an abnormal protein that can no longer function. So genomics has the potential for helping us to understand the role of those genes and the proteins that they make and how that translates into human diseases. Okay, get ready. We're about to go into protein synthesis. Now, I'll warn you, this can be a little bit sticky, so I want you to watch this particular video several times. And when you're reading this in your textbook, I want you to be really careful to focus on the content that we cover and don't go too in-depth in it right now. Okay, we need you to understand the basics of what protein synthesis is. Remember, synthesis is simply making. So we're going to look at how proteins are made. There are also some animations that you'll find in Mastering A&P that will show you sort of a cartoony type version of how proteins are made. If I can find some links for those, I may post those at a later date. However, if I don't get to it and you're ready for it, you can find some stuff on this in the student study area of Mastering. Just do keep in mind that I'll be making your test, so I know how in depth I need you to know this. All right. So let's get started. Much of the cellular machinery is devoted to synthesizing the large number of diverse proteins that we need in order to carry out our metabolism, to do and to be who we are and what we are, to make protein structures, to make body parts. Proteins determine the physical and chemical characteristics of a cell. The instructions for making those proteins, so the instructions for protein synthesis, are found in the DNA in the nucleus. Protein synthesis involves two steps. Step 1 is transcription, and it occurs in the nucleus, where we take the instructions in the DNA and we turn it into a strip of RNA. Remember, RNA is going to carry the code that is encoded for in the DNA. So the DNA has the recipe. The RNA makes a copy of it and brings that copy out into the cytoplasm so it can be translated into a protein. Okay, that's the basics. So let's begin with that first step, and remember, transcription is going on in the nucleus. So transcription is the process where genetic information encoded in DNA is copied onto a strand of RNA called messenger RNA, or shortened into mRNA, and that's going to direct the making of the protein. So besides serving as the template for messenger RNA, DNA can also synthesize two other kinds of RNA, ribosomal RNA and transfer RNA. Confused yet? Don't be. We're going to figure all this out. So let's begin by looking at transcription. Okay, remember we're inside the nucleus, and here we've got a strand of DNA, and this little yellow thing is an enzyme called polymerase that is bound to the DNA. It breaks those hydrogen bonds, so here's a strand of DNA, here's a strand of DNA, and it's no longer wound together in the double helix, but it's broken apart. So now we've got one strand of A's, T's, C's, and G's, those nucleotides. Here's the other strand of A's, T's, C's, and G's. Okay, so remember this purple one and this purple one, if the DNA was in its normal form, would be complementary base paired together. But because it's not, we've broken it apart because we need to make a protein. The sequence of these nucleotides is going to determine the next set of base pairs that comes in and forms a strip of RNA. Now remember, transcription means to transcribe the information in the DNA into a strand of RNA. So everywhere there's an A on our DNA, we're going to put in another nucleotide that matches it. Now remember, in DNA we have A's, T's, C's, and G's. In RNA we swap. That T is now a U. Okay, so here where we've got an A on our DNA, we put a uracil. Here where we've got a thiamine, we put an adenine. Where there's a guanine, we put a cytosine and a guanine, an adenine and a uracil. Okay, so this is still complementary base pairing with these nucleotides, and we're making a new strand of nucleotides that is going to be a strand of messenger RNA. Now remember, the sequence that these nucleotides are put together on the RNA is determined by this strand of DNA. Okay, so far so good? All right, so this is going to continue until it gets to the end of that particular gene. Okay, the end of the gene is like the end of the recipe. So we put all of these nucleotides together, and we've created this strand of messenger RNA, and then we're going to have an enzyme that's going to come in and cut out the pieces we don't need. So sort of like there was an extra step or an extra ingredient in the recipe that we didn't need in there, so we get rid of it. All right, so now we've got our strand of messenger RNA, and here's the nuclear envelope. Okay, remember we're still inside the nucleus up here. So this strand of messenger RNA is going to go out through a nuclear pore into the cytoplasm. Now this DNA up here is going to zip right back together until the next time we need to make more messenger RNA for that particular gene. Okay, so far so good? Okay, now our strand of messenger RNA that we just made in the nucleus is out in the cytoplasm. Translation is the process of reading that messenger RNA nucleotide sequence to determine the amino acid sequence of the protein. Okay, so transcription gets all the information inside the nucleus from that gene and makes the mRNA. Translation is translating that into the sequence of amino acids. Remember back when we talked about proteins and I said that the primary structure of a protein is the sequence of amino acids? That sequence of amino acids is determined by, let's go right back here for a moment. The sequence of amino acids is going to be determined by the sequence of these nucleotides that we just put together to form the messenger RNA. So translation happens as this. Messenger RNA comes out and attaches to a ribosome. Okay? So a ribosome is a protein structure that directs or causes protein synthesis to occur. Okay, so a while ago when I mentioned that that ribosomal RNA, that's just a part of this ribosome. Okay, so ribosomal RNA is a part of the ribosome structure. Okay, so the messenger RNA comes out and attaches to the ribosome. The tRNA, the transfer RNA, is going to bring in amino acids according to the sequence of those nucleotides on that messenger RNA. So let's move to the next slide so we can sort of see how this all works together. Okay, so we begin here at number 1. Okay, so this is a part of our ribosome here, this little pale cloud looking thing. Okay, so here's our strand of messenger RNA, and right here is a tRNA molecule. So this blue little squiggly thing and the green thing attached to it is a particular amino acid. So on that messenger RNA, we divide those nucleotides into sets of 3 nucleotides called a codon. So a codon is simply 3 nucleotides in a row on the messenger RNA. The tRNA also has, and you can see up here in this image, the tRNA also has 3 nucleotides attached to it. And nucleotides, remember, are going to always complementary base pair. So here, this codon of the messenger RNA, this AUG on the messenger RNA, is going to determine which tRNA comes in. So we've got to have a tRNA that's got an AUC, so it complementary base pairs here. Okay? Or let me rephrase that, a UAC that's going to complementary base pair with this codon. So your codon complementary base pairs with an anticodon, that's just the 3 nucleotides that are attached to your tRNA. So there are different tRNA molecules with a different set of amino acids that code for all 20 of the different amino acids. So you may have a tRNA amino or a tRNA molecule that has UAG, and it has amino acid number 1. And then you may have a tRNA that has the nucleotide sequence of CCG, and that's going to have amino acid number 2. And then number 3 would have a different one, number 4, and so on. So the sequence of your codons, remember that's your nucleotides, is going to determine which tRNA molecule comes in, carrying its particular amino acid. So that tRNA comes in, binds to the mRNA, bringing its amino acid in. Then the next tRNA molecule comes in, bringing its amino acid. Well, which one comes in? It's all determined by these these nucleotides, remember? Because these have to match to these, and these bring a specific amino acid in. Okay? So now we've got 2 tRNA molecules sitting here. Each one has an amino acid, and a peptide bond forms between those amino acids. Now, once that peptide bond forms, these 2 amino acids are hooked together. This tRNA molecule can now go away, go back out and pick up another amino acid. And then the next tRNA molecule comes in, bringing its amino acid, and the next one comes in, bringing its amino acid, and the peptide bonds continue to form between all those amino acids, forming a polypeptide chain. Okay? Now, let's think back to what we just mentioned there. This whole thing started in the nucleus with the DNA. The DNA was split apart by an enzyme. So now we've got 2 strands of nucleotides. RNA is formed in the nucleus when more nucleotides come in And complementary base pair, let's just back up. Complementary base pair to this gene and its nucleotides. So we build that messenger RNA, we snip out any of it that we don't need, and then we send the fixed version or the best version out of the nucleus into the cytoplasm. Once we're in the cytoplasm, that messenger RNA binds to a ribosome. Once it is bound to a ribosome, tRNA molecules come in carrying amino acids. Each amino acid forms a peptide bond with the neighboring amino acid. So the next one that comes in forms a peptide bond with the one before it. And that creates a polypeptide chain. Once we get to the end of that messenger RNA, no more tRNA molecules can come in. When no more tRNA molecules can come in, we're at the end of that polypeptide chain. It snips off from the ribosome and goes now to the endoplasmic reticulum, where it can be folded and modified and sugars can be added, and we can turn it into a functional protein. So sort of just a short summary of what's going on again. Remember, we're making a protein which is made up of a sequence of amino acids. Those amino acids are brought in by tRNA molecules to bind to the RNA, the messenger RNA. Okay? The messenger RNA was built because of the sequence of nucleotides in the DNA. Now, I really want you to take some time and understand this. It can be a difficult concept, but once you get the first part of it, then move to the next part, and then move to the next part. This is really important because proteins have to be made correctly in order to function correctly, because those amino acids determine how a protein is built and determine how that protein folds, and that all determines whether or not that protein works as it should. Just to give you an example, if those amino acids are not put in the correct sequence because there was a mutation in the gene, so that means the RNA wasn't built right, which means the tRNA molecules didn't come in the right way, bringing the right amino acids. So that's what happens when sickle cell anemia forms. There is a mutation in the DNA that causes the wrong transcription, which leads, or the wrong translation and the wrong transcription and the wrong sequence of amino acids. So sickle cell anemia causes the wrong protein, or is caused when the wrong protein is made. Okay, we're going to shift gears a little bit now and begin talking about replication of DNA. So replication of DNA is sometimes also called cell division. So it's the process by which cells reproduce themselves. It consists of nuclear division and cytoplasmic division. So that nuclear division is sometimes called the process of mitosis, when we're making more cells that are just replacing other body cells, or we're growing. So cell division that results in an increase in body cells is called somatic cell division and involves a nuclear division called mitosis, plus cytokinesis. So mitosis is division of the nucleus, cytokinesis is division of the cytoplasm. So that's one type of cell division. Cell division that results in the production of sperm or egg cells is called reproductive cell division and creates gametes, sperm cells and egg cells, and it consists of nuclear division called meiosis and cytokinesis. Now we're going to look at the basic difference in those types of cell division. So let's begin with replication of DNA. So here you can see we've got our double strand of DNA. We've got our nucleotides, complementary base paired to each other, attached with those weak hydrogen bonds. Okay? Those hydrogen bonds get broken, and then we have two single strands of DNA, and we're going to bring in more nucleotides and make another strand of DNA that matches with each one of those new single strands. Okay, so the purple ones are what we started out with. The double strand, complementary base paired nucleotides bound together with hydrogen bonds. We split the hydrogen bonds. Now we've got an old strand purple, an old strand purple. New nucleotides come in and form hydrogen bonds. A's to T's, C's to G's. Over here on this old strand, A's to T's, C's to G's. Now we've just formed two genetically identical strands of DNA. Keep that in mind as we move through this, okay? So the cell cycle is a sequence of events that allows a cell to duplicate its contents and divide in two. It consists of interphase and mitosis, okay? So the cell cycle is basically what a cell does in its lifespan, okay? So interphase is all this we have in purple. Mitosis is this part down here in this light pink and red, okay? So interphase is when a cell is doing its cell job. So a liver cell is being a liver cell, a toenail cell is being a toenail cell. So interphase is when the cell just carries out its metabolic processes, except for mitosis, okay? That's a separate phase. So we divide interphase into three separate phases. Now, normally a cell is going to spend about 80% of its life cycle in interphase, just being a cell, not reproducing. So we divide interphase into three separate, shorter phases. In the G1 phase, the cell is metabolically active, so it's duplicating its organelles and all the components in its cytosol, except for the DNA. Sometimes cells stay in the G1 phase for a very long time, and then they may not ever divide again. So sometimes we change that G1 phase to a G0, which means the cell is just not dividing. Cells then move into the S phase, S for synthesis, where the chromosomes are replicated. And then in the G2, sometimes called the growth 2 phase, cells grow, and the cell continues to complete its preparation for starting mitosis. So a cell in interphase shows a very distinct nucleus and the absence of chromosomes. So let's look at the next slide here. We're going to look at some cartoon versions as well as some microscopy of cells in each of these phases. So these two cells are in interphase. So you can see the nucleus here. You can see the nucleus here. You can see the cell membrane. You can see the cell membrane. And here you see that centrosome made up of the centrioles and the material that surrounds them. Okay? So in interphase, a very distinct nucleus, and your chromosomes are in the form of chromatin, which is just all the DNA all wound up into balls. It's just like if you took a skein of yarn, you know, like Granny crochets blankets and stuff. If you just took all that and you piled it into the floor and then wadded it up into a ball, that's sort of the way chromatin is when the cell is not undergoing mitosis, okay? So when you look for interphase, remember it's divided into the G1, S, and G2 phases. The nuclear envelope is still present, and the chromosomes are still all wadded up in the form of chromatin, not nice condensed chromosomes. The cell then moves into mitosis. Mitosis is when the nucleus of the cell is going to divide. It results in two sets of chromosomes being made and then separated into two separate nuclei. Mitosis consists of prophase, metaphase, anaphase, and telophase, followed by cytokinesis, the division of the cytoplasm. So let's look at those phases. The first phase of mitosis is known as prophase. This is when the chromatin condenses and shortens and becomes what we commonly call chromosomes. Okay, so here you can see chromosomes are becoming visible as compared to this cell right here. This one's still in interphase. Remember how the nucleus looked very different? Okay, so during prophase, the chromosomes become visible and the nuclear envelope starts to disappear. So here you can see a late prophase, the nuclear envelope is gone. You also see these spindle fibers starting to form and come out near the chromosomes. So the spindle fibers are going to form from this centrosome and attach to those centrioles. So the centrioles start moving to the opposite ends of the cell. Okay, so remember, prophase, the nuclear envelope disappears, chromatin condenses into chromosomes, centrosomes move to opposite poles, and the spindle fibers form. Meta means middle, so notice now where all of the chromosomes are located. Okay, so the chromosomes line up near the center of the cell. The centromeres, or the part that joins the chromosomes, is basically split right down the center of the cell, so that you've got each copy of the chromosome on opposite sides now, right? The spindle fibers are going to come in and attach to the centromeres. So here you can see in the histological or the microscopy image, all of the chromosomes are lined up and you see the spindle fibers attached. During anaphase, the chromosomes are going to split and separate, being pulled apart at the centromeres. As those spindle fibers shorten, they're going to drag those chromosomes apart, so they're starting to pull those copies of the chromosomes to the opposite ends of the cell. Okay, so here you can see that we're just beginning anaphase in this image, and over here you can see anaphase is really in progress because the chromosomes are separating into their two copies, or their sister chromatids, these are called. Okay, so the sister chromatids are being pulled to opposite ends of the cell. And then we move into telophase. So telophase begins as soon as the chromatid movement stops. The identical sets of chromosomes are at the opposite poles or opposite ends of the cell. They start to uncoil and revert back into that form of chromatin, just all wadded up. The microtubules disappear. The nuclear envelope reforms, and the new formed mitotic spindles that we use to separate the chromosomes start to go away. So here you can see the chromosomes are in two separate areas. Okay, so we've separated these out, and this structure known as a cleavage furrow forms. Okay, so a cleavage furrow makes this look sort of like an hourglass. So we're going to continue to squeeze the proteins in this cleavage furrow, sort of like wrapping a belt around your waist and squeezing yourself in two. That's what we're doing here to separate these two sets of chromosomes into two cells. So telophase, the mitotic spindles disappear. The chromosomes go back to chromatin, and the nuclear envelope starts to reform around each of those two new nuclei. Now we don't have two complete cells yet because they're still attached at that cleavage furrow. Cytokinesis is the division of the cell cytoplasm and all those replicated organelles. So that process forms in late anaphase or early telophase when we start forming that cleavage furrow. When cytokinesis is complete, interphase begins again, and the cell starts doing its cell job. Now remember, because we replicated the DNA and then divided it into two separate nuclei, we've got two genetically identical daughter cells. All right, so you need to know each of these phases and what's going on. What's happening with the chromosomes? What's happening with the nucleus? How do we know what phase is which when we view it? What characteristics identify each of these two phases? All right, so this sort of gives you just a table form of what we just looked at. So you have your interphase with your G1, S, and G2. Then you have mitosis with the phases of mitosis and what's going on inside the cell. And then cytokinesis, which separates the two cells. The second type of cell division, and we are going to cover this one very, very, very lightly, is called meiosis. Remember when we began talking about cell division a few minutes ago, I mentioned the two types of cell division, with mitosis creating two genetically identical daughter cells. Well, meiosis is the process where we create sperm cells and egg cells. The big difference in these is that where we created those genetically identical daughter cells in mitosis, each with 46 chromosomes, meiosis has to separate chromosomes because sperm cells can't have 46 cells and egg cells have 46 cells, or the offspring that they're creating would have 96 chromosomes or 92 chromosomes. So meiosis is sometimes called reduction division because it results in cells being created that only have one copy of the chromosomes. So it has two sets of division. Remember we had interphase, prophase, metaphase, anaphase, and telophase in mitosis. Well, we have that happening twice in meiosis. So meiosis 1 happens, and then meiosis 2 happens. So chromosomes are replicated and then divided in meiosis 1, and then meiosis 2 divides them again because we have to separate those chromosomes so that there are only 23 in each sperm cell and 23 in each egg cell. So remember, normal cells have 46 chromosomes, one copy of each. So meiosis separates those two copies. We replicate them, then separate them. Okay? So here you can see a little chart showing you the differences between mitosis and meiosis. Also remember that we're creating two genetically identical daughter cells in mitosis. In meiosis, we create 4 cells that are not genetically identical. All right, just to wrap up this unit or wrap up this chapter, aging is a normal process that's accompanied by the progressive alteration of the body's ability to maintain homeostasis. There are many, many specialized branches of medicine that deal with problems in the care of the elderly. That care or that branch of medicine is called geriatrics. Physiological signs of aging are gradual deterioration of function and the capacity to respond to environmental stresses. Signs are related to a net decrease in the number of cells in the body and the dysfunction of the cells that we do have. So cells don't reproduce as well, and sometimes mutations have occurred and accumulated in those cells. So when cells do reproduce, they're reproducing faulty genes that then, of course, can also lead to faulty proteins being made. Again, we'll talk much more about aging as we continue our studies, and that will finish up our Chapter 3 content.